these are some background slides which are already covered this morning. This is a slide that actually I borrowed from one of the lectures I gave with Genentech. This is a quote from an article in 1939, the American Journal of Röntgenology, which is a radiology journal. And essentially what the investigators did is that they implanted a tumor into the earlobe of a mouse, and then using photomicrographs, imaged the blood vessels that were growing into the mouse. And they basically realized that with the tumor growing, there was neoangiogenesis, and this was 70 years ago. And so this is not so new of a concept, but it's evolved over time, as you've heard this morning. And in this paper, they wrote, one is almost forced to the conclusion that there is, associated with viable tumor growth, some blood vessel growth stimulating factor. And of course, the prima donna, as was said earlier, is VEGF. So a necessary step in the progression of angiogenesis is actually, the progression of a tumor is actually the angiogenic switch, which is this concept that as a tumor grows, it can only grow to 1 or 2 millimeters, but then the switch is turned on with proangiogenic factors and inhibition of anti-angiogenic factors, and the tumor can grow larger. And that's shown here in this slide, where you have small tumors, less than a centimeter, which are not very vascularized, and then as a tumor grows, you can see blood vessels that are sort of on the surface, and then on the right panel you see a fully sort of vascularized tumor as it grows further. So these are important steps in angiogenesis, and I think we're really just at the tip of the iceberg in terms of inhibition of angiogenesis, in terms of the multiple different factors that are involved. I throw this slide up. This is from Alex Ajay a few years ago, and the primary reason for this is that the next 15, 20 years of drug development will be sort of really marred by our ability to do the right trials in the right patients. It's not that we have a dearth of new options. We have many, many new options, many new drugs. We just need to identify the right patients in which to test these drugs. And that really gets to the idea that we do need better biomarkers to select patients. This alludes to what was talked about earlier, where we can't afford to do 1,000 patient, 1,500 patient studies to look at a single molecule and look at a marginal benefit. The idea is that if we can select the right patient, we can have a dramatic benefit in a very select population. And it gets to the idea of individualized medicine, which I prefer that term over personalized care. So in gastric cancer, there have been several studies that are looking at new agents evaluating targeted therapies. So not too long ago, the TOGA study was reported by Eric Van Cutsem and colleagues, and that for the first time showed that targeted therapy does work in gastric cancer. For those of you who are unfamiliar with this is actually a very dreadful disease. Median survival for metastatic disease is less than a year. Patients who are HER2 positive who received trastuzumab with chemotherapy had median survivals of 14, 16 months, which is, quite a good outcome. And so that really led to a dawn of a new era where we were seeing many different studies evaluating different targets. So the MET inhibition studies were actually performed and ongoing, ornithuzumab and relatuzumab, and AMG 337 to name a few. There are additional evaluations of the HER2 pathway, and then what I have boxed is actually all the different approaches for anti-angiogenic therapy in gastric cancer, beginning with Bevacizumab and the AVAGAS study in the first-line setting, Rama Serum AB in the second-line study, regorafenib as well as apatinib. And so we'll talk about these studies further. The first study, as I mentioned, was Bevacizumab in combination with chemotherapy in the first-line setting for metastatic gastric cancer. This phase 3 study was actually the largest study at the time of an anti-angiogenic therapy in gastric cancer. It was built on earlier phase studies in the phase 2 setting, primarily done in the United States. Patients were randomized to receive chemotherapy with placebo or chemotherapy with bevacizumab. And although overall survival was not significant, the hazard ratio is 0.87. That's the top panel. Progression-free survival was significant, hazard ratio of 0.8, and the p-value of 0.004. And there was a 10% higher response rate, and this was with the RECIST response criteria, not any other type of criteria. So there's a hint of activity of chemotherapy with bevacizumab showing a benefit, although it didn't meet overall survival benefit. And as Professor Bertoloni explained, maybe it's a lack of a marker. If we were able to identify a marker, we could also achieve the overall survival benefit as well. So in the face of compelling phase 2 data, how do we explain the AVAGAS results? It didn't meet the primary endpoint of overall survival. The p-value was 0.1, and there was a marginal 2-month benefit in survival, but there was some evidence of efficacy with improved progression-free survival and improved response rates. So a key aspect of this study was the mandatory biomarker analysis, which was actually in over 700 patients. So 90% of the study population did actually submit their tumor sample for evaluation. And I show sort of a schematic of the VEGF pathway. We see the ligand VEGF-A, which serves as a ligand for both VEGFR1 and VEGFR2. And we see the co-receptors, neuropilins, which act as co-receptors, sort of activating also the pathway. So a potential biomarker from this large analysis, 700 patients, was plasma VEGF-A. So just to remind you, VEGF-A is the ligand. So tumors that are high in VEGF-A presumably might be more dependent on VEGF-A signaling for activity. So looking at this curve and just walking you through a little bit, the dashed lines are the tumors that were high above the median for plasma VEGF-A. And you can see that the red dashed line are patients who had high plasma VEGF-A who received chemotherapy with bevacizumab. Their survival was better than the gray dashed line, which is high plasma VEGF-A that received placebo. So it's consistent with the idea that high VEGF-A tumors were more dependent on VEGF-A signaling, and when they received bevacizumab, they came back to the norm. Whereas the low VEGF-A levels, the solid lines, they had no difference in survival with bevacizumab. Remember, everybody got chemotherapy. The other marker that was of interest was Neuropilin I. So just going back to this figure here, Neuropilin I is a co-receptor. So tumors that are high in neuropilin actually are not dependent on VEGF-A for activity. These tumors actually have VEGFR2 signaling somewhat independent of the ligand. So the converse was the idea that low Neuropilin I tumors may be more dependent on VEGF-A signaling. So here, it's the reverse. So if you looked at the solid lines that are neuropilin levels less than the median, if you received bevacizumab and you had a low neuropilin, you had a better survival than patients who received placebo and had a low neuropilin. And then conversely, people with a high neuropilin were independent of VEGF-A signaling, and there was no effect of bevacizumab. So this is summarized here in this chart, which is a little bit easier to understand. So if you look at the VEGF-A biomarker, high VEGF-A was a suggestive biomarker, and the hazard ratio is 0.72. And if you look at the neuropilin, low neuropilin was the biomarker, and the hazard ratio is 0.75. So the p-value is a p-value for heterogeneity. So it actually tests how likely is it that these two are really different. And it's borderline, 0.07, 0.06. So, and this is actually very stringent actually criteria, because if you look at just the high VEGF-A alone, it's the confidence limit, the 95% confidence limit is 0.57 to 0.93. So in and of itself, it's actually a significant finding. But in terms of heterogeneity, it's 0.07. So it requires a validation study. But this is one of the first studies, large-scale, multi-center randomized study that examined in a systematic way multiple panel of biomarkers and came up with a hypothesis that actually made some sense. So we looked at the phase 2 studies that examined bevacizumab with chemotherapy and the AVAGAS study, which is a large phase 3 study that was negative. And we looked at sort of clinical characteristics because it was still puzzling why, in phase 2 results, the results seemed so promising. And we found that actually the two cohorts were actually quite different. So gastric cancer is a heterogeneous disease. There's differences in histology, intestinal versus diffuse, tumor location. So proximal tumors behave more like esophageal cancer. Distal tumors behave more like true gastric cancers. Distal tumors are caused by chronic H. pylori infection. Proximal tumors are caused by reflux and obesity, for example. So if you look at this cohort, the U.S. cohort, more than half, 60% of the population of 150 patients were actually, in fact, GE junction tumors. Whereas on the AVAGAS study, which is a global study, half of the patients enrolled were from Asia. Only 14% were GE junction tumors, so quite different. If you look at histology, there was an even split between diffuse and intestinal on the AVAGAS study, but there was a significant more number of intestinal tumors on the U.S. cohort. And then in terms of visceral metastases, the U.S. cohort actually had, half the patients had liver metastases, whereas only 34% had liver metastases in the AVAGAS cohort. So this led to the idea that perhaps it's not that bevacizumab didn't work, we just actually had too heterogeneous of a population in the AVAGAS study. So so this was the first indication that not all gastric cancer is the same worldwide. And actually, this is an important concept that will actually be, a key featured item in ASCO this year, for example. As we approach the globalization of cancer, we have to realize that not all cancers, even from the same organ, are the same. So we look back at these biomarkers, the neuropilin level and the plasma VEGFA levels across proximal, diffuse, and distal gastric cancer, and we see some differences. Although in the blue row, that's the medians for the scoring for neuropilin and VEGFA, we see that diffuse gastric cancers were actually predominantly lower in neuropilin A, and distal gastric cancers are very high in plasma VEGFA. So we see that there may be a clue that not all gastric cancers are the same, and not only that, they may not actually have the same kind of angiophilic profile that may make you sensitive to bevacizumab therapy. So this may be an answer to the question, why was AVAGAS a negative study? So proximal tumors appear to have the worst profile for bevacizumab. Generally, they're high for neuropilin and low for plasma VEGFA. Diffuse and distal tumors appear to have at least one biomarker that supports the benefit of anti-angiogenic therapy. So I'll come to VEGFR2. So Rama Serum AB is a specific antibody inhibitor to VEGFR2. And there are many reports that suggest that this is actually the key driver for neoangiogenesis. And so here's some description of Rama Serum AB. It's a IgG VEGFR2 specific inhibitor. It blocks it at with a very low IC50 of 0.8 nanomolar, and it inhibits ligand-dependent VEGFR2 activation and signaling. It inhibits proliferation and migration of human endothelial cells, and there are mouse models that suggest activity. So with very little data, basically phase 1 data, and the AVAGAS study, the company decided to move forward with a phase 3 study, 2 phase 3 studies with evaluating Rama Serum AB in gastric cancer. The first that came out was actually the REGARD study, which you heard about a little bit earlier today. This was a 2-to-1 randomization of ramucirumab versus placebo in the second-line setting in gastric cancer. Now, just pausing here a second, this is anti-VEGFR2 monotherapy, no chemotherapy given with this, and so it's kind of bold to actually do this by itself. And I think, to some respects, a little bit of luck was involved because, in fact, the study was positive, both from a progression-free survival standpoint and overall survival standpoint. The hazard ratio for overall survival was 0.78. The p-value was 0.047. You couldn't get much closer to being a negative study, but it was in fact positive. The median survival in patients who received ramucirumab, as was shown earlier, is over 5 months, which is similar to what chemotherapy provides as well. And then the Rainbow study is the second-line study that was shown. This was chemotherapy, paclitaxel, given on a weekly basis with or without ramucirumab. And this was a little bit larger study, 1-to-1 randomization, and you've seen this Kaplan-Meier curve earlier. The addition of ramucirumab to paclitaxel improved survival up to 9 months in the second-line setting. So already you know that this is kind of a unique population, patients who are able to go onto a second-line study in gastric cancer, but there was an improvement in survival, and the hazard ratio is 0.8 and the p-value 0.02. And here is the progression-free survival, also superior, with the addition of ramucirumab. So, and then these are the data that are summarized. Response rates were improved, 28% versus 16%, and the disease control rate was also improved with the addition of ramucirumab. So multiple different measures of efficacy showed benefit with ramucirumab when combined with weekly paclitaxel in this disease. So comparing AVAGAS, the first-line study with bevacizumab, and the Rainbow study, which is a second-line study of chemotherapy with ramucirumab, there are some differences. One is a first-line study, one is a second-line study. The bar is a little bit harder to achieve with the first-line study. Both studies were very large. Together, 1,400 patients were randomized. But you see differences. So in the AVAGAS study, 50% of patients were from Asia. On the Rainbow study, a third were from Asia. And you see, in terms of overall survival, the Asian population actually did not benefit. The hazard ratio was 0.97 and 0.99 for the addition of the anti-angiogenic therapy. So it actually may be a numbers game. It may be that anti-angiogenic therapy, bevacizumab and ramucirumab work equally well in non-Asian populations. And that's suggested by the overall survival in non-Asian patients, 0.67 hazard ratio with bevacizumab in the first-line setting, and 0.73 with ramucirumab in the second-line setting. The one thing that's a little bit, this is the overall survival difference. The one thing that I would point out is actually the progression-free survival at the bottom of the page there. In Asia, the progression-free survival with the addition of bevacizumab was 0.92, whereas with ramucirumab, the progression-free survival was better. So this is the first hint that maybe the two antibodies aren't identical. They have slightly different effects, and I think we need to explore this a little bit better. And then there's a third positive study. This is a Chinese study of apatinib, which is a VEGFR2 tyrosine kinase inhibitor. This is done in the third-line setting, so not first-line, not second-line, but third-line gastric cancer. Who would have imagined? This is a 2-to-1 randomization of apatinib monotherapy versus placebo. This is the second study evaluating an anti-angiogenic as the monotherapy in gastric cancer. And once again, we have a improvement in survival with apatinib in the third-line setting with this monotherapy. And the hazard ratio is 0.71 and the p-value 0.01. Regorafenib is being evaluated in the INTEGRATE study. Regorafenib is a kind of a dirty tyrosine kinase inhibitor. It hits RAS and RAF, and is also angiogenic with hitting VEGFR1, 2, and 3, and TIE2. This is the INTEGRATE study. This is 150 patients with esophagogastric cancer, 2-to-1 randomization in the second-line setting, and we will get results of this later this year. So, to conclude, gastric cancer is more than one disease. I think this is an important message to take home, is that as we develop clinical trials across the globe, we need to know which patients we're actually including. Different gastric cancer subtypes have different prognoses. The TCGA data recently came out that suggested there are at least 4 different types of gastric cancer, and how they fare with anti-angiogenic therapy remains an open question. But based on the studies that we've reviewed today, we can firmly say that anti-angiogenic therapy is a proven target in gastric cancer, which is, I think, an important statement. There's data for Rama Serum AB, apatinib, and even Bevacizumab in the first-line setting with improvement in PFS and response rates. We still need candidate biomarkers, and I think without having candidate biomarkers, it will be an uphill challenge. And with that, I'll take any questions.
So maybe I'll start. What future trials are being contemplated now, or underway, or being thought about in terms of extending these, successes? What's next?
Yes, so great question. So there was a lot of interest in repeating the AVAGAS study, but in a, perhaps a Western population. But that wasn't pursued by Roche Genentech. Mm-hmm. With the positive results with Rama Serum AB, actually there's lots of efforts with Rama Serum AB across the panel in gastric cancer. So one thing I would mention, and this was actually brought up earlier, was the sort of dosing issue with targeted therapies. So particularly for antibody therapies in gastric cancer, there's now several lines of evidence that suggest that actually the dose of the antibody can't be translated from one disease to the other disease. So right. The TOGA study, which looked at trastuzumab, basically used the trastuzumab dose that was given in breast cancer patients. It was an 8 milligram per kilogram load, and then maintenance of 6 milligrams per kilogram as maintenance. And it turned out that in that study, a third of patients were underdosed. The trough level of trastuzumab was lower than expected. So the FDA has mandated a second study called the HELOE study that is actually evaluating the standard dose of trastuzumab versus a high dose. So similarly, for Rama Serum AB, the same thing was identified, where there appeared to be a dose effect, and about a third of patients actually did not reach a magical number of 50 micrograms per milliliter concentration. So they are doing a couple studies evaluating alternative dosing schedules of Rama Serum AB with chemotherapy, both in the first-line setting and the second-line setting, to see if they can improve survival further. So that's one area of research, and then the other area of research is actually combining Rama Serum AB with other targeted therapies.
And you said, like, the Regard and Rainbow trials were second line, correct?
The Regard, yes.
And so what's happening first line?
So there is actually there is a first line Rama Serum AB study that will open in the next 3 months internationally of cisplatin and 5-FU plus or minus Rama Serum AB. But the Rama Serum AB would be at actually an alternative schedule that was given in Regard and Rainbow, sort of accounting for this alternative PK.
Can you just elaborate a little bit on how an Asian versus, let's say, Caucasian, whatever, biology difference can be related to different results using anti-angiogenic drugs? Any evidence of differences in the disease in terms of vascularization or whatever?
Yeah. So that's what we were hoping.
Could I just had the same sort of question. I wanted you to define Asian, please.
Sure. So it's not an easy answer to that question. So there's one study that looked at comparison of resected gastric cancers from New York versus Korea. And to answer your question, Professor McVie, Asian is sort of typically Korea, Japan. China, the behavior of gastric cancer in China seems to mimic more the West than the East, than Korea and Japan. And I'll come to why that definition is sort of used. But in this study of resected gastric cancer, the first author is Strong from Memorial Sloan Kettering. It was 1,000 patients who were resected from Memorial Sloan Kettering and 1,600 patients resected from Korea. And they looked at all the known prognostic factors. So, the tumor depth, the nodal invasion, but then also perivascular invasion, tumor size, any preoperative characteristics, the type of surgery, and they ran a risk-adjusted model, and they identified that even when accounting for everything that was known, and this is just a few years ago, Asian patients had a better survival stage for stage than Western patients, and there was about a 10% improvement in survival. That's one suggestion that maybe there's difference in biology. When I give this lecture in Asia, it's actually not well received because the feeling is that it's all one disease and we're the same. But we really don't, it's not so clear. The other difference is that in Korea and Japan, they do screening. So almost all the gastric cancers that are on clinical trials from those two countries are actually secondary, cancers, not synchronous, meaning that the bulk of disease is much smaller. So in those places, the median survival is 16, 18 months. That's just with standard therapy. So if you're getting first-line therapy with chemotherapy and anti-angiogenetic therapy for like 6, 8 months, you have many other opportunities for treatment, and so it's much harder to demonstrate a survival benefit in those specific countries versus the rest of the world. So China, they don't do screening, and so their survival, so most patients actually present with a metachronous or synchronous tumor, the primary gastric cancer as well as metastases. So they actually tend to have a survival similar to Europe and the North and South America.
I thought there was a lot of data saying that Japan was quite different. I've no idea about Korea, but this whole idea of screening has been in the surgical literature for a long, long time. There are also apocryphal stories about groups of Japanese in parts of Japan who take 5-fluorouracil sprinkled on their cornflakes.
So it's interesting. So
because of the high incidence, and there's definitely environmental issues to do with how the Japanese get stomach cancer.
No, absolutely.
But I don't know anything about Korea. So what was the split, Japan-Korea?
Equal, actually, in terms of the Avogadro study. So just with regard to that comment, there are studies that look at individuals who emigrate from Japan to the United States, and their risk of developing gastric cancer in subsequent populations is actually in between. So there's both a genetic factor and an environmental factor as well that plays a role. And there's also pharmacogenetics as well. So for example, the main drug that is used in Korea and Japan for treating gastric cancer is not fluorouracil, it's S1. And they actually can give a higher dose of S1 than is tolerable in the West. And so it may be that's actually an important factor here.
I wanted to have a little more details on the biomarkers. I know less, VEGF, whatever, various polymorphisms, but the neuropilin was very interesting. And so, it's like there is a sort of inverse correlation with the levels. So I wanted to know the mechanism of that. And then the other question is, what is in the context of HER2 positive gastric cancer, which I think is about 20% or something, is there any combination with like lapatinib or, the anti-angiogenic drugs, for instance?
So, great question. So I'll take the second question first. In the AVAGAS study, we retrospectively looked at the patients who were HER2 positive. So it was about 10%, 70 of the 700 or so. And there was no difference in outcome with regard to survival. Of course, most of those patients did not get trastuzumab. There is an abstract that will be presented at ASCO looking at the combination of chemotherapy with trastuzumab and bevacizumab in HER2-positive gastric cancer in the first-line setting, and the overall survival on that phase 2 study is actually quite remarkable. We were talking earlier at lunch, and in breast cancer, it sounds like the combination of anti-angiogenetic therapy and HER2-directed therapy wasn't so positive, and so. I don't know where that's going to go, if that's going to move forward. There is lots of interest in combining them, but most of the studies now, to date, will exclude HER2-positive patients because these patients, about 10 to 15%, will get HER2-directed therapies, that in terms of drug development. And in terms of the biology of neuropilin and why that was a positive biomarker, unfortunately, with the negative study and the sort of Genentech was hit with several negative studies at the same time, because at the same time that AVAGAS resulted, there was also the adjuvant colon data in 2 studies were also negative. They haven't pursued actually that aspect of understanding it. So I don't, beyond sort of what's in the literature with neuropilin as a co-receptor for the VEGF receptors, I don't have more mechanistic understanding of that. Perhaps Dr. Kerbel does.
No, not much, but I think about the you mentioned the Herceptin plus
Anti-angiogenesis.
yeah, in breast, and it didn't do so well. I think it's the Beth trial. I can't remember, but wasn't that trial an adjuvant trial? That combination.
Yeah, I'm not.
There was
sure.
a phase 3 trial where, and I think it was chemo plus Herceptin plus Bev versus no Bev in the other arm, and I don't know, or yeah, something along that. But the addition of the 2 targeted agents, it didn't give a benefit. But again, I think that was an adjuvant trial.
So it'd be hard to.
And so that could explain the difference, because again, as we discussed earlier today, in the adjuvant setting, these drugs just haven't done very well. One last question. Going back to the first line trials that are underway, given the success of Rama Serum AB as a single agent, second line, do these trials, do any of them then, are they going to involve a maintenance phase? In other words, you give chemo plus Bev, but then a maintenance phase afterwards of Bev alone? Not Bev, Rama Serum.
Ram alone, yes.
Yeah.
So, so that idea was sort of debated throughout. So there isn't sort of agreement across the globe of maintenance therapy. So to do an international study, what the compromise was made was it would be about 5 months of platinum 5-FU plus or minus ramucirumab, and then the platinum is dropped, and so then people can go on 5-FU or capecitabine plus or minus ram, and after a year people can drop the capecitabine. So it's kind of a staged maintenance approach.
Okay.
I'm not sure we'll learn much from that, but
Hi. A very quick question about the group which had a high VEGF and the low neuropilin. Have you correlated that back to the enhancement characteristics on the CT? Because I'm assuming most of these patients would have had a CT
scan.
arterial phase.
Yeah, that's a great question. No, I don't think that that's been done. On the AVAGAS study, it wasn't a central radiology review, so it would be hard to make that kind of correlation. But that's a great question.
Any studies in India?
None of the current phase 3 studies, AVAGAS, ramucirumab, Regard Rainbow, the MET inhibitor studies actually included India as a as a potential site. I think it's a lost opportunity. There's many patients, as you know, with gastric cancer in India, and it'd be interesting to see how they would do.